So this was on molecular configuration. And from this point and on, we focus our discussion of amino acids on their side chains. If we look at this table, we see the 20 types of amino acids that normally appear in proteins. They are called the standard amino acids. There are also two other types, but they appear in a small number of proteins, so they're not included here. And we can see that each amino acid has a full name, but also a 3-letter and 1-letter abbreviation. And if we look at the last column, we see the organic groups that compose the side chains of the standard amino acids. And it's very apparent that they're very versatile chemically. We have different groups here, like aliphatic chain, aromatic chains. We have alcohol groups, amides, amino groups, and so on. But still, we can classify all the 20 amino acids to only 4 groups. And the main feature, the main parameter by which we classify them is their polarity, or the polarity of their side chains. And according to this classification, we have non-polar amino acids, which means amino acids whose side chain is non-polar. And we have amino acids whose side chains are polar, and they can be electrically charged or uncharged. The fourth group is classified not by polarity, but by the inclusion of aromatic rings, 1 ring or 2 rings in the side chain. And this is a more specific parameter, but again, most of the amino acids are classified according to their polarity. We already discussed polarity in the introduction section, and we saw that this property results from differences in electronegativities between atoms that are connected through chemical bonds. And indeed, when we look at the side chains of polar amino acids, like we can see here, in this case, uncharged polar amino acids, we see that virtually all of them contain bonds between an electronegative atom like nitrogen and oxygen, and another atom which has a lower electronegativity like hydrogen, or as in this case, carbon. So in all these cases, the difference between the electronegativity of nitrogen or oxygen and that of the other atom creates a dipole. And this dipole is actually what makes this bond polar, and makes the entire chemical group polar. And we can see above, here we can see the units of electronegativity in each case. And above, we can see the difference in electronegativity between the nitrogen or oxygen and the other atom connected to it. So in all these cases, you can see that the electronegativity difference is above 0.8 units. And we consider this number as a threshold for polar molecules. If the difference between the more electronegative and less electronegative atom is above 0.8 units, then we consider the group to be polar. Now, we have one example, we have one case in which this is not what happens. We have an amino acid called cysteine, which is considered as a polar amino acid, but its side chain contains a bond between sulfur and hydrogen. Now, if we look at the electronegativity units, we see that the difference between them is only 0.38 units. So this means that this group and the whole amino acid cysteine shouldn't be polar, but it is polar because sulfur also contains 2 lone pairs of electrons, and this puts a negative charge density on the sulfur and makes the bond more polar than it should be if we just consider the electronegativities. On the other hand, we have amino acids whose side chains are non-polar. And when we look at the chemical groups composing their side chains, we see that they contain only bonds between, well, in most cases, bonds between carbon and hydrogen atoms or between carbon atoms. So in the second case, it's easy to understand why such a side chain is nonpolar, because the electronegativity difference is 0. But also in this case, where a carbon atom is bonded to a hydrogen, the difference in electronegativities is very low. It's only 0.4 units. And that's because the electronegativity of carbon is also low and similar to that of hydrogen. So all of these side chains are non-polar, but we have one case, that of the amino acid methionine, which contains also a sulfur atom. Now, when we talked about polar amino acids, we saw that when a sulfur atom is bonded to a hydrogen atom, then the bond is polar. Here, we have a different case. The sulfur is bonded to two carbon atoms, and this whole group is considered to be non-polar because of two reasons. The first is that the difference in electronegativities is small, but more importantly, the two carbon atoms face opposite sides of the sulfur, which means that the very small dipoles that do exist here and here cancel each other out. So all of these groups are considered to be non-polar. So why is polarity so important that we use it to classify amino acids? There are two reasons for that. The first one is that it underlies the non-covalent interactions which drive protein chains to fold into three-dimensional structures and allow them to interact or to bind their ligands. So in the case of protein folding, for example, the favorable interaction between non-polar amino acids and the unfavorable interaction between these amino acids and the surrounding water molecule is driving the chain to fold into different structures, in all of which the non-polar amino acids are buried inside the core of the protein. In this way, they are shielded from the surrounding water. And in this image, the non-polar amino acids are shown in yellow. And the polar amino acids, which are here in green and blue, are mainly on the surface of the protein, where they can favorably interact with the surrounding water and contribute to the solubility of the protein. So that's at least what happens in proteins that reside in water-based media, such as the cytoplasm or the blood. The second reason for the importance of polarity is that it also underlies chemical reactivity. And this is important in, for example, in enzymatic catalysis. Here we can see one of the most common mechanisms of catalysis, in which a nucleophile attacks a carbon atom, forms a covalent bond with it. There's another group that leaves the molecule, and the reactant becomes a product. So what allows the nucleophile to attack the carbon atom in the first place is that both of them are polar. They just have the opposite polarity. The nucleophile has electrons on its outer surface, which makes it partially negative. And the carbon atom is the opposite. It has a partial positive charge because it's part of this dipole with oxygen. So the empty orbitals of carbon receive the electrons of the nucleophile and form the covalent bond.